Welcome to Travel. Today we are going to talk about flow cytometry basics in our biology and life sciences lecture series by Dr. Abid Ali Khan. That's gonna be me. I have a PhD in molecular biology and biomedicine, have almost a decade experience of teaching and research at different universities around the world. So without any further delay, we are going to start today's lecture about flow cytometry, and this is going to be one of the first lectures about flow cytometry that we are going to talk about in the next few days, starting with the basics. So today's lecture will be focused just on the basics. Let's start and let's discuss what is flow cytometry. So flow cytometry is a powerful technique. It's basically an analytical technique which is used in biology, especially in immunology and in clinical diagnostics. In other cases, for example, in industrial microbiology and other processes also, flow cytometry is quite oftenly used these days. It's a powerful technique, it's an analytical technique which sort individual cells or particles. Initially, the technique was discovered and started with particles, but now a number of applications are performed with cells. So basically what happens is the analysis performed by flow cytometry for particles or cells, they are based on the physical and the chemical characteristics of the concerned particles, and it is used for the analysis of optical as well as fluorescence properties of the sample, which could be in the form of cells or it could be in the form of particles. And then the analysis in terms of cells would not only be talking about the morphology, which is actually the size rather than the morphology, but as well as the internal complexity of the cells, in this case the organelles if it's a eukaryotic cell, and if it's not a eukaryotic cell, then what's inside the cell or what's inside these particles. So flow cytometry applications, there are many applications of flow cytometry as we discussed already in clinical diagnostics. For example, hematological disorders, flow cytometry is one of the phenomena that is really deeply connected to blood analysis. Conventionally, blood is always and used an example to discuss flow cytometry. In the research, flow cytometry also have a lot of applications. For example, in the gene expression, how much DNA content is there, and in the industry, for example, in beverages industry or some other industry where the viability of cell is playing a big role. It's used for cell viability and vitality. And why is flow cytometry used instead of the conventional microbiological or molecular biological methods? The reason is it's cost effective, it's way more faster, rapid analysis, and last but not the least, it's efficient and it's accurate, because any cell or any particle that is in the sample has to go through in front of the laser, and if it goes through the laser, it cannot hide from the laser. So as we know in the figure number 1, we can see that there's a laser light, an incident laser light, which actually is going to hit all the samples coming in a streamline under the laminar flow. And once these particles or these cells are exposed to this laser light, there would be scattering, which is divided into two scattering. side scattering as well as forward scattering. Just simply telling you forward scattering in terms of flow cytometry, we'll talk about the size of the particle, and side scattering would tell about the internal complexity, or in some simpler terms, how complex is the internal organization of a particle or a cell. So the bigger or the larger the size of the particle, the more the forward scattering we would see, and the more the internal complexity of a cell or a particle, the more the side scattering. An example would be if you run a bacterial cell, which is pretty small in comparison to, let's say, a eukaryotic cell, and you would see if the forward scatter is plotted on the x-axis, and let's suppose on number one, value number one. So bacteria is if bacteria is at 0.1 in the forward scatter, a eukaryotic cell would be much bigger. Let's suppose it will be 6 at the value number 6 or value number 7, whatever units you are using here. These are just intensity units. Similarly, on the side scatter, it will end up way over bacteria, because bacteria's internal complexity is pretty simple. It's rather, there is only a nucleoid region, there is no membrane bound nucleus, and there are no cellular organelles. So that's how we talk about, or how we refer to, the internal complexities. So also continuing this, we know that flow cytometry is also used for fluorescence. When the laser hits a cell or a particle which has been conjugated with a dye, depending on the emission, the excitation spectra, the fluorophore or the dye would absorb the energy and release its emission, and then the detector with specific mirrors and optics and different combination will detect this particular emission photons, and then tell you by a combination of different diodes and PMTs and stuff, which electronics will tell you if this particular cell is present or it's not present. Okay, so continuing this, we will talk a bit about the basics of flow cytometry. After the introduction, I would just focus briefly on what are actually the principles of flow cytometry. One of the major principles of the flow cytometry is the hydrodynamic focusing. This means that the sample and the sheet fluid, so we can see that the sheet fluid is going to be running along the analysis all the way through. So when we perform the analysis, the sheet fluid, which is a fluid, has to be running and generating this laminar flow, which will make the samples run in a stream of single cells. So the sample and the sheet fluid, they would converge to ensure a single cell line or single particle line, which will make the particles go through the flow cell at this complete structure that you can see in the figure above, in which the sample is injected and then it's run in a stream line of single cell, is called flow cell. Next is the light scattering, as we discussed here, the side scattering and the forward scattering. So the cells or the particles, when incident laser light is hitting them, they will scatter the light, and this scattering will give us information about either the size of the particles, that's the forward scattering, or the complexity or the granularity of the sample, which is the side scatter. Last but not the least, in simpler terms, is the fluorescence detection. So as we see in the figure here down, in the figure that is below here, we have a flow cytometry analysis figure with dye tagged cells, and these cells can be labeled with any fluorescent dye or with antibodies, and to the antibodies there would be attached some dyes, and this could be with, of course, capturing for specific primers. And then, of course, there's an excitation laser, excitation and emission spectrum. So the laser, for example, if it's a blue laser with a 488 nanometer wavelength, then it will hit the dyes, and the dyes will absorb energy, and then there will be an emission spectrum, and the emission spectrum will have its own wavelength, and based on these two wavelengths, we can detect many of these particles. Talking about forward and side scatter, so you could see in this image to the left, the first image to the left, we have a cell and we have an incident light source. In this case, it's a laser light. Cells are very sensitive to light, so they will mostly absorb. Majority of the light would be absorbed. Some of the light would pass through, but there would also be cases when some of the light incidents would be deflected, and this deflection depends upon the internal complexity of the sample or the cells or the particles. In this case, we can see that this is the cells have a nucleus. So when these rays hit, when the laser hit the nucleus, there will be some deflection. Now we could see that the incident light source would detect the size by forward scattering. So this would be the forward scatter. And then with the deflection, we could find out the internal complexity. And the more the internal complexity of the cell or the particle, the more the side scatter would be. And one thing is very important here, that the side scatter detector is always at a 90 degrees angle to the cells. So doesn't matter how many at how many angles the light is deflected, the detection would only be with the for the incident laser beam that is deflected with an angle of 90 degrees. On the right side of this slide, we see a classical example of flow cytometry, where the whole blood of eukaryotic organism, let's suppose it's human beings, is analyzed, and you could see there are three different populations denoted or represented in this graph: in green, in red, and in blue. So we know blood has lymphocytes, monocytes, and granulocytes. Lymphocytes, the B or the T lymphocytes, they are the smaller in size and the simpler in complexity, and this is why they're ending up here at 200. Now monocytes are a little bigger, larger in size than lymphocytes. That's why the forward scatter, they would appear bigger. So you could see they are roughly at 400 on this x-axis in the forward scatter. They also have a little more internal complexity. All the immunology experts would know this. That's why they are appearing a bit above, so a bit higher on the scale on the side scatter. So on the y-axis we have the side scatter, and on the x-axis we have the forward scatter. Granulocytes, for example, eosinophils, basophils, all these have a lot of internal complexities, and they are also relatively larger in size in all three of them. Though the size between monocytes and granulocytes, there's not a lot of difference. That's why you could see on the forward scatter, that is the x-axis, there is not a huge difference where the monocytes and the granulocytes, and they are more or less in the same region. But on the y-axis, the side scatter, the granulocytes are way more up on the scale, because they have a lot more granularities, these particles. So let's continue. The next slide is a fluorescence flow cytometry. And as we discussed, it's one of the powerful techniques in biology and medicine for the analysis and the quantification of individual cells or individual particles in a fluid suspension. To detect markers on different cells or tissues, or cells of the tissues, or even in particles, flow cytometry can be a wonderful technique. And fluorescent dyes are commonly used in this case. They are also known as fluorophore or fluorochromes, or just dye. And in this example, in this particular example, we have two cells. So we have cell A, which is light blue in color, and then we have cell B, which are reddish maroon in color. And we have these antigens on the cells. You could see they are like these semicircular structures. We have antibodies for both. So this antibody would with a fluorophore with a red star. That red star means this is a dye. And then we have another antibody with a blue star, that again with a blue fluorophore. And this antibody, first antibody would respond to cell A, the antigen of cell A, and the second antibody would respond to and bind to the cell antigen of cell B. Now, when we incubate them, this is very important. The sample preparation is very important in terms of flow cytometry. So we have to properly know about the incubation, how long, which temperature, light is needed, dark conditions are needed. These are all very important. And the antibodies will attach. Now, the next goal is, when we have proper incubation, we want to know in a mixed sample of antigen of cell A and cell B that are treated with antibody A and antibody B with different fluorophores, we can find out how many of cell A and how many of cell B have these antibody attached to these antigens. This could be interpreted. We want to see how many cells A and how many cells B have these antigens present on these cells. So it could be an example of diagnostics. If this antigen is present on cell A and cell B, this means either the presence of a particular cancer or any other pathological condition. And what we will do, we will run them, and let's suppose these two are dyes that are excited by red laser. Both of them are excited by red laser. We will use a red laser flow cytometry, also flow cytometer analysis, and we will plot in a way that in this case you can see on the x-axis we have plotted the emission. So now this is the emission. It's very important to understand. The excitation would be the red laser itself. The wavelength of the red laser itself is in the region where these fluorophores would absorb the red laser as an energy source and then get excited. And then when they are excited, the detectors would detect the excited states, they detect the wavelength of the excitation, and we will plot the blue fluorophore excitation or fluorescence in this case on x-axis and the red on y-axis. And as you can see in this particular x versus y histogram, that we have the presence of both cell A as well as cell B. This means in this particular example, in this mixed culture, in this mixed population of cell A and cell B, both of these cells contain these antigens. If these antigens would not be present, the antibodies would not bind to them, and we would not see the fluorescence, because the fluorescence is coming from the fluorophores attached to the antibodies, and the antibodies would only be present if they find this particular antigen in this cell. And we can see 10 to the power 4 is the fluorescence of blue cell, and more or less same or a bit more for the red cell. This is a classical example. In reality, it would not be like this. There will be so many more things. For example, in this quadrant, you could see that there are a lot of background. So in the next video, I will discuss about going detailed on fluorescence, excitation, emission, and then giving some example of different fluorophores, how do they end up, and I will also discuss about the dot plots of flow cytometry to make you understand well. So if you like this lecture, if you would like to have more lecture on fluorescence and flow cytometry, please give us a thumbs up, like the video, share the video, and mention in your comments on our lectures. Make it way more interactive with you guys. Please subscribe and see you soon in the next lecture video. Thank you. Take care of yourself. Have a good time. Bye bye.